and they are very different. Maybe they only have few things in common, but all in all, they are different organisms. So for each group, we are going to talk about the characteristics that they share in that group, their laboratory work, what they look like in the laboratory, and how they are cultured, and we are going to talk about their clinical relevance. In other words, the human infections associated with them, and then we'll talk about the investigations and a little bit on antimicrobial management. So we'll start sharing. I have very many screens now. You can see it. Yeah, this. So are you able to see the screen?
Yes.
Oh, okay. So the organisms we are covering this morning, there's the genus Corynebacterium, and then the genus Bacillus, and then there's a group called Actinomycetes, which has several genera in it. So we'll start with Corynebacterium as a genus. The organisms there are sometimes referred to as Corynebacteria. But in that group, the bacteria are gram-positive, rod-shaped organisms or bacilli. And they are not typical bacilli. They show a lot of variation in shape and size. So some are very small, some are almost round. Occasionally, one might see a typical bacillus, but all in all, they vary. They show a lot of variation in shape and size. The organisms in that genus, Corynebacterium, have swollen or club-shaped parts, a club-shaped part at the end. So they are referred to as club-shaped organisms by some authors, just to describe what they look like under the microscope. We have talked about variation in shape and size. The other property they have is that they stain irregularly. It's the nature of the cell wall that allows the stain to come out more in some parts than others. So they show irregularity in staining. They are organisms which don't form bacterial spores. They are not motile, and they grow as facultative anaerobes. So in that genus, there are many species. Among them, there's one significant species, which is associated with a severe human infection, and it is Corynebacterium diphtheria. And it's the main pathogen among the species. Then there are many other species, they have their specific names. And from time to time, they might be implicated in some infection, but it's not common. And they are lumped together just to separate them from Corynebacterium diphtheria. They are referred to as diphtheroids or coryneiform organisms. So we'll elaborate on Corynebacterium diphtheria because it's the main pathogenic species, and it illustrates most of the characteristics shown by the members of that genus. So they have that characteristic of variation in shape and size, and that is described as pleomorphism. And when they are gram-stained, they show the club-shaped ends. So the class rep, in case you lose me, you communicate to me by phone. My internet sometimes fluctuates. Can you hear me?
Yes, we can hear you.
Yeah, just once we lose connection, you let me know, then we stop and look for it. But you do that by phone, I'm having it nearby.
Okay.
Yeah. So the Corynebacterium bacteria, they are pleomorphic, gram-positive bacilli with club-shaped ends. And they have some specific staining properties, just like what we have mentioned. For the genus as a whole, they show that irregularity in staining. In addition, they are also very sensitive when it comes to gram staining, to the step where the colorization is done. So they easily lose the first reagent, the first stain in the gram staining reagents, the one that the organisms which are gram-positive would retain, it easily loses it. So they easily get decolorized in gram staining procedure. And if it loses it, then a counter stain is added. It is usually neutral red or sapphire, whatever it is that is being used, it is what it will take. So when the smear is examined after staining, the organisms might look like they are gram negative. But all in all, they are gram positive, and they are gram positive, and those who handle them, they know exactly how to apply the decolorization process so that they don't lose their gram positivity. Another characteristic they have is that they contain granules in the cytoplasm. All bacteria have granules here and there in their cytoplasm, but the ones for Corynebacterium bacteria are more pronounced, they are more common, and they are a feature that is useful when it comes to their identification. For the other bacteria, the granules are there, but they are not significant. They might remain there for some time and disappear, so then it's not a consistent feature. But for Corynebacterium bacteria, the granules are important, they are significant, and they are consistent. And these granules are referred to as metachromatic or volutin granules. And they contain some metabolite. That metabolite has a scientific name, but it's good to know that they are made of they are storage form of the metabolite of the organism. And these granules tend to be concentrated at the ends of the bacillus. That swollen end contains a lot of them, a lot of these granules.
Dr. Odhiambo, your voice
Yeah.
is a bit low.
My voice, it is that is my maximum sound for the laptop. So we can't go further. Let me just confirm that. Yeah, it's maximum. It can't be made louder. So is it too low for you to hear?
No, as in right now, it's okay, but sometimes when you're teaching, your voice goes low.
Oh, okay. Maybe the position of my, yeah. Okay, I'll try to keep it at one point. So the last characteristic I wanted to talk about is their typical microscopic morphology or appearance. Under the microscope, when these organisms are stained in their typical state without much disruption, maybe in some material taken from a lesion where the infection is going on, most bacilli occur as pairs. So many of them are thin, paired, and they are attached at one end. So when you have two bacilli attached at one end, they form an angle. So they are angled. They are described as angled pairs. Then there are others also. Many of them are arranged in groups, but parallel to each other in groups. And that arrangement is referred to as palisade arrangement. So those are the two major characteristic features of the microscopic field, what one would see. But in addition to those two, there are others which are randomly placed, singly, or many, so that it gets mixed up, and what is seen has been described as showing Chinese letter alphabets, or Chinese letters, or Chinese alphabets. Because both the paired ones and parallel ones and the other random ones, they all occur in one field. And they are observable, all of them, and the picture is said to resemble the Chinese letter alphabet. But for our practice, most of what we do would be from a culture, and it would be smeared or spread out, so you are not likely to see the typical arrangement. Significant physical properties of Corynebacterium diphtheria. It's an organism that is capable of retaining viability for weeks in dust or on dry surfaces. And this property is important when it comes to transmission. The longer an organism stays somewhere outside the body, the higher the chances of a susceptible host getting into contact with it, and the higher the chances of it being transmitted to another person. So that point is relevant when it comes to transmission. And then as other bacteria, it is susceptible to heat and regularly used chemical disinfectants. Then we described it as the main pathogen in the group. So it has some properties that would enhance its pathogenicity. And these are mainly that ability to be transmitted from one person to another. That is important. There are some organisms that are not transmissible from one person to another. But for this one, it can be, and we have said it could be even from contaminated places. Secondly, its ability, it has the ability to invade mucosal surface locally at that part where it is attached to, the part where it infects, it now invades and colonizes it and multiplies there. And that is significant when it comes to pathogenicity. And then lastly, and more importantly, it's an organism that produces an exotoxin. And that exotoxin is referred to as the diphtheria toxin, and this is its major virulence property. It's the property that is responsible for tissue damage and some of the manifestations that are seen in association with the organism when it infects a human host. So the main virulence property, diphtheria toxin, it is responsible for the severity of the disease as other virulence properties, but it's not released by all members of the species. You have a species containing quite a number of bacteria, referred to as Corynebacterium diphtheria. They are meeting the characteristics required to belong to that species, but they are not able to release the diphtheria toxin. Sorry, they are not able to, we are talking about the toxin, to release the diphtheria toxin, so they are not able to cause the illness that is caused by the illness where the toxin plays an important role, and that illness is diphtheria. So I hope I've explained it well. So it is a toxin released by Corynebacterium diphtheria, and not all members of the species are able to produce it. And then we are going to learn later in later moments, or in a few minutes' time, that the illness associated with this diphtheria toxin is called diphtheria. So not all members of Corynebacterium diphtheria cause that illness, diphtheria. So the reason why the other members are not able to produce it is because that ability to produce it is acquired, it is developed. And it is developed only as a result of genetic material acquired from bacteriophages. So only those members of the species that have been infected by those specific bacteriophages are able to produce it. The ones which haven't been exposed to it are not able to produce it. So the specific bacteriophages contain the genetic information for the synthesis of the toxin. And this transmission of genetic material through bacteriophages, and specifically that one of production of diphtheria toxin, might have been touched on when you did bacterial genetics. So it's a process of gene transfer via bacteriophages, and it's specifically referred to as lysogeny. They become lysogenized. And Corynebacterium, because of that, in that species, the species can be divided, can be classified further, and it is classified into toxigenic strains, the strains which are capable of producing the diphtheria toxin, and then the non-toxigenic strains. And these non-toxigenic strains lack the ability to produce the exotoxin. So it is the toxigenic strains that cause diphtheria. So the clinical significance of Corynebacterium diphtheria, we touched on this, but just to put it together, one is the illness diphtheria as the main illness caused by this organism. And when it occurs, it's an acute disease associated with outbreaks. Obviously, it starts with one person, but then it spreads until it becomes an outbreak in a community, and it affects children more than adults. And the source of infection, maybe the one who passes it on to the first patient, maybe a nasal carrier or a patient, another patient. And the bacilli are transmitted via secretions or droplets from the upper respiratory tract of the patient or the nasal carrier to the upper respiratory tract of the susceptible person. So once it gets into the upper respiratory tract on the mucosal surface, it multiplies there at the site and releases the exotoxin. And then that toxin acts on the mucosal surface cells at the point where it is released, and it also diffuses and enters the blood circulatory system, and it goes to the other parts of the body. Otherwise, the organism does not go further than the area, the site of infection, the mucosal surface. It remains confined to that part, and it does not invade into deeper tissues, and it does not invade into the blood circulatory system. So the diphtheria as an illness, how does it manifest? Initially, it is confined to the oropharynx, and that is the upper respiratory tract and the part close to the mouth. And the manifestations are due to the local invasion of the mucosal surface by bacilli. And because of the presence of the bacilli, the body responds by mounting an inflammatory process to try and eliminate the infection. And also at that site, the organism multiplies and produces the bacteria toxin, which acts on the cells at that surface. So you have the bacteria toxin acting, and we have the inflammatory response also. And all that is in the mucosal surface at the site of infection. So generally, the person will have fever, like other infectious diseases. And then specifically, if one looks at the throat of the affected person, it's associated with inflammation, which is observable as a grayish membrane-like covering. Is somebody saying something?
No.
Okay. So it's seen as a grayish membrane-like covering, and because it is like a membrane, but not a membrane. And it might look grayish to some people, but it is referred to as a pseudomembrane because it's a false membrane. What it is made of is a lot of things, products of inflammation. That's some fluid that comes out of that site because of inflammation and damaged cells, the tissue, the mucosal surface tissue cells, and the inflammatory cells. They are all get suspended, caught up in that fluid, and that would form a layer covering the site where the inflammatory process is going on. So that is what looks like a membrane on that surface. Then externally, if the infection is very severe, the neck would look swollen in front. And that would be due to the inflammatory that is going on in the inside part. And that would cause edema, and sometimes maybe the lymph nodes of the neck would be enlarged, but would be maybe contribute to a lesser extent to the appearance, the external appearance. So the neck looks swollen, it looks full. Some people have described it to resemble a bull neck. So you'll see in the textbook if you read the bull neck appearance. Another manifestation that is another, is it a manifestation? Another problem, yeah, it is a manifestation. It would be difficulty in breathing. And this is because all this is happening in the respiratory tract, the airway, where the air passes through. This inflammatory fluid, the fluid, the edema, causing the edema, the pseudomembrane, they are all happening on the airway and can be very extensive sometimes, but all in all, the manifestation is difficulty in breathing. So they are more in association with what we have described. This would occur as complications, but they can also occur as other effects of the diphtheria toxin. One of them is the airway obstruction. We have talked about an airway that is threatened by an inflammatory process and its products. So if it is very extensive, sometimes it can obstruct the airway. So airway obstruction is a possible complication that is usually looked for, and should it arise, then it's managed as quickly as possible. Secondly, through the blood circulation, we said the toxin gets into the blood circulatory system. And via the blood, it reaches various tissues, and there it may cause damages depending on the tissue. Among the tissues affected is the heart muscle. Sometimes it can cause the heart muscle, and that would compromise the function of the heart. Sometimes they present with heart failure. Sometimes they develop abnormal heart rhythm. Secondly, the tissue that can also be affected is the peripheral nerve. So it depends on which peripheral nerve, but whichever one it is, it causes degeneration and affects the function of that nerve. And thirdly, the adrenal glands, they can also be damaged by the diphtheria toxins activating in the blood, and that would impair the release of the hormone that it's responsible for, and that would affect the functions of that hormone. So that's the main illness associated with diphtheria. And then secondly, as a pathogen, it's capable of causing wound infection. So for wound infection, it's uncommon. It can infect an open wound. It's been isolated from time to time, but it's not a common causative agent of wound infection. And when it occurs, it's any species, any member of the species. It doesn't matter whether it's capable of producing a toxin or not, toxigenic or non-toxigenic, it would cause wound infection, because wound infection is through contamination of the wound and then local invasion, and it's not associated with diphtheria toxin release or action. So for laboratory investigation of diphtheria, the specimen would come or would be obtained from that site, the upper respiratory tract or the oropharynx. And it can be the fluid, or it can be, that is the exudate from the inflammatory process. It can be infected tissue in that part, and it's all part of the pseudomembrane itself. What is important is to take it with care. It's a delicate area, but in addition to that, it's important not to introduce other organisms in that part. So one uses sterile techniques, sterile instruments, and other sterile methods that are necessary. Then the procedure is basically demonstrating the presence of that organism in that specimen. And that is done first and foremost by gram stain. One can make some prints or smears, smear from fluid or the prints of that material, express it on the slide and do a gram stain. And we have mentioned, if it is not disrupted very much, one might, if they are there, one might be able to see the gram-positive bacilli showing the various arrangements that we described. And because it's tissue or from an infectious process or inflammatory exudate, one also expects to see other contents. Those inflammatory cells, dead tissue, apart from the organism. Then it's isolated for identification because we are talking about the bacteria itself. So one needs to isolate it and identify it. So to isolate it, you need a culture medium, and here the commonest culture medium in any bacteriology laboratory is useful. It has adequate nutrients. Serum is very nutritious for bacteria, or whole blood that would provide adequate nutrients for the growth. The only setback is that the specimen is coming from the upper respiratory tract, and there are many, many other organisms in that part. It's one part we say contains many organisms as normal flora. And they are also capable of growing on the air. So one will have the organism growing and many other organisms growing, and it will be very hard to handle. But nevertheless, it's inoculated just in case, or whatever is causing, it might have some use, just in case at the end of the whole exercise, one finds that the infection might not be due to Corynebacterium diphtheria. And if it is due to any other organism causing that severe inflammatory process, then it will be captured that BA. But specifically for Corynebacterium bacteria, one needs a selective medium, a medium that would suppress the growth of those other many unnecessary organisms and enable the Corynebacterium bacteria to grow and be identified. And so the selective medium is recommended for primary isolation of Corynebacterium bacteria. So it's possible one needs to inoculate the two media at the same time. So the commonly used selective medium is actually BA, but it has a chemical added to it to make it selective. And the chemical is potassium telluride. So it is referred to as potassium telluride blood agar, abbreviated as PTA. So it suppresses the potassium telluride, suppresses the growth of other organisms in the specimen, and enables Corynebacterium bacteria to grow. And as it grows, it reduces telluride. And the product is dark in color. So that product is incorporated into the colonies, and the colonies appear dark in color. So there will be some shade dark gray to black colonies on a medium that looks like DA. Although we have said it has potassium telluride incorporated in it. So that will become clearer during the practical illustrations. Another medium that is relevant when it comes to Corynebacterium bacteria is called Loeffler's serum agar. This particular one is not used for isolation. It is used after the organism has been isolated, and its place is to help in the identification of the organism. So it is used after primary isolation in the process of identification, and it is a medium which is rich in nutrients. It carries sugar, carbohydrates, whatever is needed, and it facilitates rapid growth. And since it's very rich nutritionally, the organism takes up a lot of those nutrients and ends up forming a lot of volutin granules. So volutin granule formation is quite marked. And it grows very fast after it has been isolated from the specimen. It's inoculated here, and within 6 hours, it has grown and formed the colonies that can be analyzed. So the colonies from it are analyzed for the presence of volutin granules, just to confirm the identification. And these are demonstrated using some specific staining methods. One of them is called Albert stain. There's another one called Neisser stain. Again, this would be illustrated in the practicals. What now the volutin granules in the cell look like when they are stained. So during isolation, whether it's primary isolation or in Loeffler's serum agar, the samples are incubated in air. We say they are facultative anaerobes, and they grow well at 35 to 37 degrees centigrade. And for the primary isolation, it can take 18 to 24 hours to form colonies which can be studied. But for Loeffler's serum agar, we have said within 6 hours, the colonies are well formed, and one can demonstrate whatever needs to be demonstrated from them. So based on what was seen in the specimen, based on what has been grown on PTA and then Loeffler's serum, one is quite convinced that one is dealing with Corynebacterium bacteria. So growth characteristics would help also in confirming the identification, and also colonial morphology. We have talked about the color they take when they grow on PTA. Then after cultures, the colonies uh, are stained. Gram stain is made from the culture. Again, one wants to have consistency, gram-positive, geomorphic bacilli, and then alpha staining is done, especially from the low flow serum measure. And then biochemical tests, there are some, again, you'll be told what they are in the practical. They are applicable where needed, and they help sometimes to differentiate the toxigenic strains from the non-toxigenic strains. Even those toxigenic strains can be divided further depending on the severity of the illness they cause, and to get those further divisions of the toxigenic strains, again, there are some biochemical tests which might be needed. And lastly, for one to call the illness diphtheria, it is important to demonstrate the ability of that isolated organism to produce the diphtheria toxin. And there are many tests in the laboratory which are used to demonstrate that. And again, that part, there's one called the ELEC test, and there are other ways to read about it. It will be demonstrated in the practical. So one needs to confirm that the organism isolated from the throat of that patient is capable of producing the diphtheria toxin, because that is when it will be called diphtheria, because it's associated with a toxin. But one can still have that kind of respiratory tract infection by the strains which do not produce, the non-toxigenic strains. So when it is like that, then it's not diphtheria. It's just upper respiratory tract infection due to Corynebacterium diphtheria. And one cannot call it diphtheria. So what we are saying is that diphtheria is an upper respiratory tract infection, but it's associated with the toxin, or the diphtheria toxin. But you can still have upper respiratory tract infection that is associated with Corynebacterium diphtheria, but it's not associated with the diphtheria toxin. So it will just remain as upper respiratory tract infection due to Corynebacterium diphtheria. So we are following up the diphtheria, the management methods of diphtheria. Specifically, one needs to give the antitoxin, the antibody to the toxin that is being released, that is being spread via the blood circulatory system, and that is causing tissue damage at the site. So the antitoxin is administered, and then antimicrobial agent treatment to clear the organisms from the infection site. And among the agents, penicillin and erythromycin. So if one has the upper respiratory tract infection without the toxin, one would still give these antibiotics and they will still be infected. That is specific. But for the other manifestations, they are managed accordingly. Where the heart is affected, that is now beyond the organism. It's further, it's managed in other disciplines by other people. Then supportive care is necessary, and one significant area where this would become necessary would be the upper respiratory tract. If there is airway obstruction, or if it's almost obstructed, then whoever is taking care of the patient needs to create a method for enabling the patient to breathe. Then there are measures of prevention and control, being something that spreads within a community. And one such measure is detection and treatment of carriers and patients. Then management of patients in separate rooms from others. And then active immunization. This Diphtheria toxoid is given to children as part of the immunization process. And it is believed that when the routine immunization programs were launched, since that time, the diphtheria as an illness has become less and less. It's rarely encountered. Another point when it comes to control measures or prevention regards the population that is affected. One can examine that population to detect who is susceptible and who is not, who is not protected and who is not protected. So there's a test that can be done. It's a skin test to detect those who are predisposed and those who are not. One wants to detect whether they've been exposed to have the antibody to the toxin. That is the, So if they are not, then they would not have it. So the idea is that if one is found not to have it, then they can be immunized so that they develop the antibody. And that test is called Sheik test. So when you come across it, then you will know what it's all about. That it's part of the control measures in the community, try to protect those who are not yet affected and can be affected by knowing who they are and immunizing them so that they develop the protection.
So,
Then the diphtheroids. These are organisms which are very similar to Corynebacterium diphtheria. They occur as saprophytes in various parts of the environment, and they can also cause normal flora, or they are part of the normal flora on the skin and mucous membranes of the nose, of the throat, and lower part of the urinary tract. And they are commonly encountered as contaminants of specimens and cultures. We said they have names. They can be identified and they can have their specific names. But all in all, what is important is that they are differentiated from the pathogenic, the main pathogen, the Corynebacterium diphtheria. Otherwise, as other normal flora and saprophytes, they're capable of causing infections or diseases when an opportunity arises. So they are encountered uncommonly as causative agents of opportunistic infections. So that is the first genus, the Corynebacteria. So the second one is Bacillus. So the genus Bacillus, under the microscope, the members are gram-positive rods, but these ones are typical. They are clear-cut rods, and they can be seen arranged in chains or singly, one at a time. And this is a genus of bacteria that forms spores. They are spore formers. They form bacterial spores, which are also called endospores. And most species in this genus are motile. Most of them, that means there are some which are not, and among the few that are not, Bacillus anthracis is one of them. It's non-motile. So we are going to mention the species. One of the species will be Bacillus anthracis. So this particular one is non-motile, and then majority of the species in that genus form colonies or in BA, a medium that contains intact red blood cells. And when they grow on it, they cause hemolysis. they lyse the red blood cells around the colonies. So they are surrounded by complete zones of hemolysis, hemolytic zones. That is the majority. Again, there's a small percentage that doesn't do that, and Bacillus anthracis is one such organism. It is mostly non-hemolytic. And then they grow in air as aerobic organisms, and they can also grow as facultative anaerobes. So we'll again pick on Bacillus anthracis later, but if we look at the species in general, that Bacillus anthracis is the main pathogen, and then there are other specific species which have their own names, and then there are other species, many of them collectively referred to as anthracoids. Again, just to separate them from the main pathogenic species. So among other species that are named or commonly encountered among the ones which are not Bacillus anthracis, we have Bacillus cereus, there's Bacillus subtilis, Bacillus humilis, and Bacillus thermophilus. So we say a few more things about Bacillus anthracis. Being the main pathogenic species, it's important for that pathogenic characteristic for us to know about it, but it also reflects most of the characteristics of the organisms in that particular genus. So under the microscope, it's typically gram-positive bacillus or rods, bacilli when they are many, and rods. And they can be seen under the microscope as stained film or smear as single cells, or one cell attached to another to form chains. So the morphology of stained bacilli in a chain is likened to a bamboo rod. So those of you who have seen bamboo, there are nodes of bamboo. So I guess each node would represent one bacillus. So you hear about bamboo rod appearance or resemblance. The organism, when it's causing an infection in a mammalian, an animal, a human being, it has a capsule. So it is capsulated in tissues. And the Bacillus anthracis capsule is a polypeptide capsule. It's not like the polysaccharide capsules that are common. This particular one is made of polypeptide, and the organism is non-motile, and it forms spores during existence outside the animal host. So that would be in the environment, and that also would be in the artificial culture when it's grown artificially in a medium. But the spores enable resistance to adverse conditions and also changes that would otherwise make the other bacteria non-viable. It's able to withstand that or to resist that and survive for a long time. So it has significant physical properties, like when you have the bacilli, that is the organism that is growing and dividing, of vegetative bacteria, they are destroyed by heat like other bacteria at 60 degrees centigrade for 30 minutes. But where there are spores, we have already talked about the resistance of the spores. These are relatively resistant. They are typical bacterial spores, and bacterial spores are formed to enable the organism to withstand difficult conditions that would otherwise render the bacterium non-viable. So the spores of Bacillus anthracis are relatively resistant to regularly used methods of treatment of items to reduce chances of transmitting infections during use. These are relatively commonly used disinfection methods. The organism tends to resist most of those when it is in a spore form. So it resists many chemical disinfectants at commonly used concentrations or recommended conditions, and some methods based on heat at various temperatures. It's capable of surviving in the environment or in animal products longer than other bacteria. So being the main pathogen, again, its important properties when it comes to pathogenicity, that ability to form spores is important because we have explained this, it facilitates transmission. It enables this organism to survive somewhere in the environment or on a surface for a longer period, and that would increase its chances of a susceptible host getting into contact with it and of it getting transmitted into that person. So the ability to form spores facilitates transmission. Then the capsule. Although it's not a polysaccharide one, it functions like any other capsule. And the capsule here inhibits phagocytosis like other capsules. That would enable the organism to invade tissues or to spread within tissues. And thirdly, it also produces an exotoxin. And the toxin is Bacillus, anthracis, and exotoxin. This particular toxin is made of 3 different components or molecules or whatever you want to call them. And these 3 components, they have names. And you can find out what their names are by reading about it. But each one of the 3 has its function in pathogenesis, and they work together for the overall function of the toxin. So it is the major virulence property responsible for the tissue damage and severity of associated illness. Usually when you have these different components, there's one that facilitates entry, there's another one that does something, and there's another part that is responsible for the tissue damage that would be important for the severity of the associated illness. So you need to know what the 3 components are and what each component does before one ends up with tissue damage and severity of an illness. So the clinical importance or implications of Bacillus anthracis. It is an organism that has been there for many centuries. The people started medical microbiology or bacteriology. They studied this organism, they worked with it, they treated the infections by it. So it has been there for a long time. And the illness associated with it is called anthrax. And this is primarily a disease of animals. Among the animals, the domestic animals can get it, also the wild animals. Among the domestic animals, cattle can be affected, sheep can be affected, and even goats. And human infections are accidental. Most of them are due to humans' close association with animals. Could be cattle herder, could be livestock farmer, or could be somebody who works in a slaughterhouse, or handles animal products. Because of that close relationship, with one way or the other, humans get exposed and we acquire the organism from these animal products. And at animal level and also at human level, the illness occurs as an outbreak. One gets an outbreak of anthrax. So we'll confine ourselves to the human illness and their possible sources and the methods of transmission of the organism to humans. So most of the time the transmitted agent is the spore. And the spores come about from bacilli. These bacilli originate from a sick animal or a dead animal. When they are sick, they produce a lot of fluid through the orifices, the mouth, the nose. They have diarrheal stool, and they pass out the bacilli into the environment, and those form spores. Spores are formed from the bacilli that are passed out by the organism, by the animal, the infected animal. Through various openings, we have mentioned the nose, the mouth, or via feces is the immediate environment. So that immediate environment can be soil, it can be pasture. So soil and pastures get contaminated with the spores. And humans and other animals are accidentally exposed from the contaminated sites. Then in addition to that, there are other possible sources of infection, and the spores can also be acquired through handling contaminated animal products. Among them, we have the skin. Let's say there are some people who trade, they sell skin or buy animal skin for one reason or the other, or process it. It becomes leather, I think, at some stage. So all those people who are handling it, there's an increased risk of getting exposed to the spores. Also bones and wool, starting from shearing it, those who shear sheep, pack the wool, and maybe deal with it at the factory level, they are all at a higher risk of getting exposed, and it can also be acquired from carcasses. If one is handling the carcass of an animal that has died, or by eating meat from infected animals or dead animals. So some people might see an animal that is sick, slaughter it and feed on it, or sell it, or call people to eat. And some of the illnesses of the animals, there are many illnesses. An animal can be sick from many other illnesses. But some people don't want to waste the animal, so they want to make use of its meat while it is still alive, so it's slaughtered and sold. And those people, it might be an illness that is due to Bacillus anthracis in that animal. And all those people doing the slaughtering or eating or handling might be exposed. So there's an increased risk of exposure for people whose occupations involve handling of animals and animal products. So among them, herdsmen and livestock farmers, dealers in livestock, hides, skin, workers in factories or places where animal products are processed. So when they are exposed, how does it get into the human body? Most of it can get in through contamination of damaged skin or mucous membranes. So the damage that would allow a bacterial spore or bacteria does not have to be a big damage. It can be a shallow bruise or very minute tear on the skin or mucous membranes. And then secondly, it can get into a human being through inhalation. It can be in the air or dust that is being inhaled. And thirdly, it can get into ingestion. And we have talked about the circumstances that can lead to ingestion. So the manifestations of human anthrax are initially, it will depend on the mode or method of infection. We mentioned the three methods. So starting with the contamination of damaged skin or mucous membrane, one would end up with a type of human anthrax called cutaneous anthrax. And this manifests as an ulcer, which develops at the site of entry where there was that small tear that got exposed to the spore. There would be inflammatory process, and it would end up with that swelling that contains some fluids initially. And then eventually it would burst and become an ulcer. So the ulcer develops at the site, and more commonly it's seen on exposed parts. These are the parts that are likely to get into contact with the spore. That it could be somewhere on the face, somewhere on the arms, or somewhere on the legs. And this ulcer is also referred to as malignant pustule. And clinically, it develops a dark part at the center, and that part is referred to as an eschar. Then the second manifestation is that which follows inhalation. So it is referred to as inhalational anthrax. It's also called Woolsotter's disease because there's some olfactory where a certain type of disease was detected to be more common about infecting the lungs, kind of pneumonia or pneumonitis, with the severe manifestations, and it was affecting, it was sorting wool in those factories. So it was given a name, Woolsotter's disease. So it is inhalational anthrax. The spores are inhaled with dust or from contaminated items, including wool. And it manifests as a severe lung infection, as I've said, a pneumonic process associated with fever, and difficulty in breathing and cough may be productive. They might cough out bloody sputum that would actually contain the bacilli. And thirdly, the type of illness acquired through ingestion causes what is referred to as gastrointestinal anthrax. So this is acquired mainly by eating contaminated meat from infected animals. Maybe it's not cooked properly to destroy the spores, or the way it is handled, but somehow it is connected with contaminated meat. And the manifestations would involve would cause abdominal, it would be, one of the manifestations is abdominal pain, vomiting, fever, and it's associated with diarrhea, which may be bloody. Then apart from those that have clear-cut methods of entry, there's also another type that is described as septicemic anthrax. It can be detected on its own without a clear-cut previous gastrointestinal or inhalational or skin one. It can just be detected as septicemic anthrax. But most of the time, it is seen as a complication of the above three. So they tend to be complicated by septicemia due to Bacillus anthracis. We say the organism is capsulated and it's able to invade tissues and invade into the blood circulatory system. So it causes septicemia. But what is significant, whether it is detected at that level, or it starts from the skin one, or inhalational one, or gastrointestinal one, it predisposes to spread of infection to other internal tissues. So one will get other tissues like meninges and other parts, they can also now become infected, and one can have anthrax that is related to those other parts. So for laboratory investigation, one needs to relate to examine the specimens and identify the organism in the specimen. So the specimen will depend on the manifestation. We mentioned a lot of manifestations, but among them we have the fluid or pus or exudate from cutaneous lesions. We have talked about septicemia, so blood would also be an important specimen. And we have also mentioned that the lower respiratory tract, you'll get sputum. So, and the gastrointestinal one, it depends on what one has. And sometimes it might even be necessary to analyze what was eaten, if it is still accessible, to compare it with what one will find in that patient. But all in all, whichever specimen one is dealing with is highly infectious and requires special techniques in collecting the samples, transporting it, and also processing it. All this is done with as a higher risk specimen, and there are some recommended protection methods where such an illness or where Bacillus anthracis is suspected. And those should be followed closely. So examination procedures, as usual, one needs to show the presence of the organism in a specimen, and that is done by staining and microscopy. So it could be fluid or pus. One can make a smear of cutaneous lesions. If one has materials from there, one can make some smears of it on a slide and stain and look for the organisms in that material. Gram-positive bacilli that we have described. But because it's a specimen, now one would see also some other materials from the tissues, some inflammatory cells. So one just needs to look out for the gram-positive bacilli. And then there are some places where they do a special staining method for the Bacillus anthracis capsular material in tissues. We have said Bacillus anthracis is capsulated in tissues, and there's a staining technique. I guess it will be demonstrated for the Bacillus anthracis capsular material. Then culture is performed. It's not a fastidious organism when it comes to nutritional requirements. It grows on the basic media, nutrient agar, and it can also grow on the commonly used medium, BA. So it's incubated in air. We said it's aerobic or facultative anaerobe, incubated at the usual temperature, 35 to 37 degrees centigrade, and within 18 to 24 hours, it has grown well enough for one to examine the culture. And the colonies, they are described as grayish opaque with a wavy margin. This wavy margin is not easy to see in our labs. You might need some magnification, maybe magnifying glasses, etc. when looking specifically for it. It's wavy margin with small projections, and the colonies are described as medusa head colonies. They resemble some hairstyle of medusa. I don't know whether medusa was a lady or a goddess, but there was that hairstyle that had some projections on it. They are relatively large in comparison to other colonies. They measure 2 to 5 millimeters in diameter. The surface is rough, and for Bacillus anthracis, most of the time they are non-hemolytic. And then gram stain is performed. The idea here is just to confirm that what has been grown microscopically looks like what was seen in the specimen as gram-positive bacilli. And we have described the various appearances under the microscope. So for identification before one pronounces it Bacillus anthracis, you have to take many factors into consideration. What was seen in the specimen, the clinical information, and in the lab conditions required for growth, and then appearance of colonies on various media. We have described the colonial morphology, gram stain morphology from the colonies, and motility test, just to differentiate it from the other species of anthracoids. Bacillus anthracis is non-motile. And then biochemical tests, should this be necessary? Then there are quite a number. One of them is gelatin liquefaction test. And one might have an idea that one is dealing with Bacillus anthracis, so much so that one might not need to do the biochemical tests. Then for antimicrobial susceptibility, there are quite a number of antimicrobial agents which are effective against Bacillus anthracis, and some of them are useful for treatment, and there might be differences which antibiotics work better for which type of anthrax. But all in all, generally among the antibiotics, we have penicillin. It's susceptible to penicillin. Just connect to power. And ciprofloxacin is also effective, as well as doxycycline. But as I said, it's not limited to those ones. It's a bigger, a longer list than that. Then the aspects of prevention and control of human anthrax. Again, we have to look at the animals because they are usually the ones that transmit the organisms to humans. And all the measures that are taken against human anthrax will be more successful if the illness is managed at the animal level. So one needs to contact the various authorities that deal with animal disease, and where there's an outbreak, they have to manage their part so that chances of human exposure to anthrax will be markedly reduced. But all in all, generally for humans, the contamination or sterilization of animal products should be applied before they are used. And then meat from ill or animals dying of infections, which are not known, or even if they are known, that meat should be discarded. There's a proper way. Those doing public health or work, they know how to get rid of these dead animals, and people should not consume that kind of meat. In isolation of patients, should there be people who are ill, they are managed in separate environments from the other patients. And we say that it's an highly infectious condition, so the appropriate protective measures are taken. Then vaccination for those known to be at higher risk of exposure. It's not something that people are vaccinated against on a regular basis, but there is room for vaccination, and there are some vaccines which come up once in a while that might be useful. They are known, risk is known, exists. Then anthracoids, these are basically saprophytes. In other words, they are found free living, they could be in your laboratory, they could be where specimen is placed. So they occasionally cause contamination of cultures. They are different from the main pathogen, Bacillus anthracis, in that they are motile. And should they be responsible for an infectious process, they don't have that capsule. Then some species that are also significant, one of them is Bacillus cereus. And this is important as a causative agent of foodborne gastroenteritis or a type of food poisoning. It contaminates food, maybe food that has been eaten and left without being covered properly or being refrigerated, left over overnight, and being a saprophyte like other anthracoids, it will contaminate the food and grow in the food. So the manifestations are due to effects of its toxins. There's a toxin that it would release in the food, but there's also another toxin that would come out once it's in the gastrointestinal tract. And it's been associated more frequently than with consumption of contaminated fried rice. Yeah, it's this more frequently it's relative. When it's encountered, it's more commonly associated with consumption of fried rice. That maybe has been left overnight and covered, so gets contaminated, and occasionally mixed. Then the other species that are important, a number of species that are used to monitor the effectiveness of various sterilization procedures. We have some methods that utilize Bacillus humilis. There's another method that utilizes Bacillus subtilis, their spores, and also Bacillus stylothermophilus is useful in that way. So Bacillus stylothermophilus spores, they are used in testing the sterilization process by the autoclave. So that must have been covered in the sterilization and disinfection. How they are used, how they are prepared, and what should happen if the method has been effective, and what should not happen, or what should happen when it has not been complete. So that's Bacillus, and we move on. You can take 5 minutes break, then we go to the next one, Actinomycetes. 5 minutes break. Is that okay? Are you hearing me?
Yes.
Oh, okay, 5 minutes.
Can we have a break?
Yeah, I'm telling you, you are not hearing. I was telling you to take 5 minutes break. Or what were you going to say?
People are requesting for 10 minutes.
10.
Yes.
Okay. Yeah, you can have 10 minutes. Okay, 10 minutes. Okay.
Dr. Odhiambo, we can resume.
What is happening? So are you back?
Yes, we are here.
Oh, okay. So the actinomycetes, microscopic morphology and description. These two are gram-positive bacteria, but morphologically, they are narrow filamentous organisms with or without branches. So sometimes these filamentous bacteria fragment into shorter forms. But when they do that, they are like bacilli, short bacilli. But all in all, they are filamentous bacteria with or without branches, and they can be demonstrated in specimen smears. They can be demonstrated in tissues and also in cultures. They are not motile, they are not capsulated, and they do not form spores. They do not form bacterial spores. And there are some of them that cause human infections. And when they do that, they grow in those tissues that they are infecting and intertwine into each other, being filaments, and they form what we refer to as microcolonies. That's a growth of the organism, intertwining into each other in tissues, and these are observable. If one looks at the infection and with a discharge associated with them, one might see some small round particles, which are referred to as grains or sulfur granules. We might have different colors, but they are microcolonies of the causative agents of those regions, and these causative agents are species or bacteria belonging to Actinomycetes group. So to mention the specific organisms we are referring to, we need to classify them. And these bacteria, they possess characteristics of bacteria because of their filamentous nature. I'm saying this because of their filamentous nature. They were actually, in the very early books, they were described as fungi, but they were studied more, and they were actually found to contain characteristics of bacteria. So their classification is like what bacteria is, how bacteria are classified. They are based on what bacteria, the characteristics of bacteria that are used in classification. So we can base the classification for convenience on atmospheric requirements for growth. And I believe you are yet to cover, I don't think we have covered any of your group, but there are some atmospheric requirements for bacteria. When you started the introductory lectures, you were told that they grow under different atmospheric contexts, the gases. So if we base it on that, there's one group that is classified as anaerobic actinomycetes. And in this group, we have organisms which are inhibited. They don't grow when they are exposed to oxygen. In other words, when oxygen is part of the atmosphere in their immediate environment. So oxygen influences their growth negatively, so much so that some are totally inhibited. So these are classified as anaerobic actinomycetes. But it's a large group again. There are some which can withstand a little bit of oxygen and maybe do better with increased carbon dioxide. But all in all, oxygen affects their growth negatively, so they fall into this category of anaerobic actinomycetes. So their degree of intolerance to oxygen varies. And when there's that variation, you have two extremes and several in between. So in this group of anaerobic actinomycetes, one genus is called Actinomyces. And in that genus, most species are encountered as part of the normal flora. You might have mentioned it somewhere, and we are doing normal flora. And they are found in mucosal surfaces in various parts, including the mouth. They are found in the female genital tract, the lower part where the normal flora are found, and they are found in the gastrointestinal tract. So in this genus, there are many species. One of them is Actinomyces israeli. That is the name of a bacterium. It has nothing to do with the country or anybody. It is just its name. And it's the main species associated with disease. Although it occurs as normal flora in those parts we have mentioned, it is associated with disease possession from time to time. In addition to that, there are also other species which are also visit in the mouth as normal flora. One is Actinomyces odontolyticus. There's Actinomyces meslindae, and there's Actinomyces viscosus. And then there's a third species which is found in animals, but it's comparable to Actinomyces israeli in humans, because we say that Actinomyces israeli is associated with disease, and Actinomyces bovis is also associated with disease in animals, and the kind of lesion that this disease is associated with is called lumpy jaw. It looks similar to what Actinomyces israeli causes in humans. So that's genus Actinomyces. Another genus of anaerobic Actinomyces is Bifidobacterium. And one of the species is Bifidobacterium gentium. And the Eubacterium, another genus of anaerobic Actinomyces, and the species here is Eubacterium nodatum. So these last two genera, that is Bifidobacterium and Eubacterium, they are normal flora in the mouth as well, and they are potential positive agents of opportunistic infection. Then another grouping is aerobic actinomycetes. And these particular ones grow in air, and organisms which are usually able to grow in air are organisms that grow in the presence of oxygen. Some of them actually require it for growth. So the aerobic ones are encountered as saprophytes in the environment. They are free living organisms in the environment, and there are many genera. Among them we have one genus, Nocardia. So the genus Nocardia is a typical actinomycete, staining as gram-positive filamentous cells. But in addition to that, it can also be stained using another method of staining that is called zero neurofilm staining technique or method that is applied when one is dealing with another genus called Mycobacterium. Maybe you have covered it, maybe not, but the Nocardia species, they also take up the stain that is used for those organisms. And when the organisms are stained using Ziehl-Neelsen staining, the results are expressed as acid first or non-acid first. And for Nocardia, they are acid first organisms. So they are gram-positive organisms, and they are also acid-fast organisms. There might be some modifications required to the original Ziehl-Neelsen staining, but still, what is important is that one can demonstrate their presence using the Ziehl-Neelsen staining, and one will see organisms which are acid-fast. So as a genus, Nocardia has several species. We have just mentioned a few of them. One of them is called Nocardia brasiliensis. This particular species is associated with infections, one in immunocompetent humans as a regular pathogen. So in that context, it is regarded as a regular pathogen. But on the other hand, it's also associated with infections in immunocompromised people. And when it causes infections in immunocompromised people, because of their immunological status, it is an opportunistic pathogen. So it is a regular pathogen as well as an opportunistic organism. Then there's Nocardia asteroides. The A for that Nocardia brasiliensis, I think got deleted. So these Nocardia brasiliensis is A, and the Nocardia asteroides. These two is a species of Nocardia. It is less virulent, and when it's encountered as an opportunistic organism. And then a third one is Nocardia cervier, again an uncommon causative agent of opportunistic infection. So those are three examples of Nocardia species that we wish to mention this morning, but again, there are other names. Another aerobic genus, aerobic Actinomycetes genus is Streptomyces. There are many species here again. One of them is Streptomyces somaliensis, and this we'll mention later in connection with SABC. But in addition to that, there are many other species. Some of them, specific ones, are non-the producers of antibiotics naturally. So initially when antibiotics came into existence, they were discovered as products of these organisms. The antibiotic can be effective against other bacteria or fungi. This particular genus contains some organisms that produce some products, natural products, which act against other microorganisms. So for our purpose today, we'll be more interested in Streptomyces somaliensis. Then a third aerobic genus is Actinomycetes. The species here, for examples, Actinomycetes maduri and Actinomycetes pelletiae. So there are many organisms. That is why we are looking at them at the level of family Actinomycetes. So we are just going to talk about most of their characteristics in general. We can't go through them one by one. And generally, in the laboratory, if one wants to grow them, we have talked of the atmospheric requirements when we are classifying them. There are some which can't grow in air, can maybe require oxygen or withstand oxygen. And there are some which are inhibited by oxygen, so they require free environment, oxygen-free environment. And then there are some among the ones we mentioned as anaerobic bacteria. They might require additional 5 to 10% carbon dioxide for better growth. So where there's a specimen where actinomyces is suspected, one needs to incubate under these three different environments or atmospheres, because one doesn't know which one is living. Then culture, one needs a medium. And commonest medium again, DA, is important here. It contains adequate nutrients and suitable. These nutrients are suitable for growth of Actinomyces species, but it can also support the growth of others, but it's particularly important for the Actinomyces species. Then there are also other media which can support the growth of various actinomyces. Some of these media are used elsewhere in bacteriology and mycology. So among these other media, Muller-Hinton can be used in some instances. Medium, Saborol's medium, or Saborol's dextrose medium is a medium that is used to support the growth of fungi, and it can support the growth of some of these organisms. And then there's a medium thioglycolate medium. This again is used to grow strict anaerobes, strictly anaerobic bacteria, which you are yet to cover, and it can support the growth of some of those anaerobic actinomycetes. And fourthly, there's a medium called Lowenstein-Jensen medium, which you are yet to encounter, that is used to grow mycobacteria, and it can also support the growth of Nocardia. So this particular bacteria grows slowly. So that is another characteristic that we share with fungi. They are slow growth, although this one is not as slow as some fungi, but it's relatively slow in comparison to the pyogenic bacteria we are used to that grow within 18 to 24 hours. This one will take 48 hours at least, but it can go on for a longer duration. Then when they grow their colonies, again we are approaching this from a general point of view, the appearance will depend on the genus, on the species, on the strain, and the growth media. But generally, some produce dry, leathery, folding colonies, which are comparable to some fungal colonies, to find some fungi. Then there are some which form colonies which are adherent to and piled above the medium. Some of them resemble breadcrumbs, but on top of the culture medium. Then there are some which look like waxy colonies. Some are powdery or chalky. And there are some that produce pigments. It may or may not be pigmented. And if there is a pigment, the color depends on the organism and the medium used. And the actinomycetes are also capable of growth in a liquid media. This is usually dispensed in a tube or a bottle, and when it grows in that, it's observed as a thin film on the surface of that fluid medium growth or aggregates on the sides or at the bottom of the tube. So whichever colony one uses, there are laboratory manuals that describe the colonies of various species, and if one has found a colony that resembles one of them, then one trains up from there. Include the gram staining, and then there's some standard bacteriology laboratory methods which are listed that one can follow until one reaches the identification of that particular actinomycetes. So the human infections associated with actinomycetes, these are uncommon infections. And then from that, we have talked about many organisms. So the infections or the lesions or the diseases they cause would be different depending on the organism. But the ones among the ones which I encountered, if they are encountered at all, most of them are associated with diseases which involve subcutaneous tissues. And these are lesions which take several months or even years to develop. And some eventually produce manifestations which are comparable to lesions associated with some fungal infections. So laboratory tests are necessary for these ones to differentiate the causative agent, whether it is actinomyces or whether it is a fungal agent, as a causative agent. So those are the ones which are encountered relatively more commonly. If one is going to encounter, one is more likely to encounter these infections that involve the subcutaneous tissues. Then secondly, there's a specific or some specific nocardia species. These ones produce or cause acute infections occasionally that would involve the lower respiratory tract. So this is different. It doesn't take a long time like the subcutaneous ones. It's acute and it involves the lower respiratory tract, that is the lungs. So all in all, the diseases can be grouped, and the main groups one is actinomycosis, another one is nocardiosis, and the third one is actinomycotic mycetoma. And all these are related to the manifestations 1 and 2. And we are going to see which one is in the subcutaneous group and which one is this respiratory tract, lower respiratory tract infection. So if we start with actinomycosis, the most significant causative agent is Actinomycetes israelii. Significant in that it's associated with most of these lesions of actinomycosis. But other species can also be involved, or other bacteria and other factors may contribute in the causation. This Actinomycetes israelii is an organism that is part of the normal flora in the human mouth. So there are many other organisms there that could be involved when in a lesion that is causing, and also other factors that go on in the mouth that can contribute. So actinomycosis initially is a painful swelling that is associated with pus formation, and that would mean it's a acute inflammatory process initially, but it takes long and changes gradually, and eventually it becomes a chronic inflammatory process. And that will involve with cause a swelling of some sort due to the processes, whatever takes place in a chronic inflammatory process. Scar tissue, fibrous tissue, etc. So actinomycosis may present as one, a lesion that involves the face, and unlucky. And that type of actinomycosis is referred to as cervical facial actinomycosis. And cervical here refers to the neck, and facial refers to the face. So it's somewhere around the mandibular area that covers part of the neck and part of the face. And they are predisposing factors to it. Some are known, some are not known. Among the ones which are known are traumatic injury of the mucous membranes of the mouth and infections which can involve a tooth or teeth. So when it starts, it involves the mucosal lining of the mouth, and then it spreads to surrounding tissues and outwards. So that is when now it comes to the external. And gradually it develops into chronic swelling, which becomes firm or hard or nodular, and it's palpated, and that is described as lumpy. So its other name is lumpy jaw. It also has another name. Those of you who are curious might end up knowing a third name, but just two are enough for us today. Then there's actinomycosis also, which can involve the lungs and chest wall, and this is referred to as thoracic actinomycosis. And this particular one starts in the lungs. How the organisms get into the lungs, uh, maybe from the mouth or extension from the cervicofacial one, but it starts in the lungs and spreads outwards, and eventually that involves the thoracic wall. And the third type is actinomycosis involving abdominal tissues, and that is referred to as abdominal actinomycosis, and can manifest as a swelling in the abdomen or pain involving a specific organ. Again, it develops slowly. Eventually, it will involve the abdominal wall. So the organism being present in those inner tissues in the female lower genital tract, it has also been associated with some pelvic infections. But the major ones are those three we have covered, but can also infect pelvic tissues. So that is actinomycosis. That is the group. Then we say the second group, actinomycotic mycetoma. So this actinomycotic mycetoma and the one we have just finished, the cervicofacial one, or actinomycosis, they are the ones which involve the subcutaneous tissues. So actinomycotic mycetoma is a chronic infection involving the skin and tissues below the skin, and it also takes years to develop. Clinically similar condition is caused by specific fungal species, which you will encounter when you do mycology. And most cases of actinomycotic mycetoma involve the foot. And this would be related to the transmission. Maybe it occurs in people who are exposed, they walk somewhere and they get pricked by thorns or sticks containing mucositive agent. So the area that is most frequently affected would be the foot around the ankle joint. And the causative agents are the aerobic actinomycetes, which we say are saprophytes, and they are introduced into the tissue through traumatic injury or traumatic inoculation. So the aerobic actinomycetes, we mentioned them. Streptomyces cerevisiae is one causative agent of actinomycotic mycetoma. Also nocardia, nocardia brasiliensis, and other nocardia species, as well as actinomadura species. And actinomadura species we mentioned is actinomadura madure and actinomadura hectiae. So those two, once they develop, they involve the subcutaneous tissue, they take long to develop, so the causative agents are different. So their manifestations now uh, become similar if you look at the lesion, although the site might be different. So the manifestations of actinomycosis and actinomycotic mycetoma, they are both chronic inflammatory swellings, which take several months or years to develop. Then when it is actinomycosis, it would involve the areas we mentioned, the jaw, the face, the hypophyseal part, or the thoracic part of the abdomen. And if it's actinomycotic mycetoma, the foot or the arm or wherever it is, but wherever it is, the skin overlying the swelling is characterized by multiple small abscesses which become eroded. And when they are eroded, they break up and form multiple small discharging wounds, which are referred to as sinuses. So it takes a long time to develop a bit of these, and as it goes on, some of them heal, some of them recur, some of them receive scar tissue, some are almost healed, some are beginning, so a mixture of them. But what is important about the multiple small discharging wounds, they extend deeply into the tissue. And what they form are referred to as sinus tracts. And out of those is a fluid that flows a discharge. And that discharge is what contains the microcolonies we mentioned at the beginning. The microcolonies of the causative agent, which are seen as small particles of various colors, which are referred to as granules or sulfur granules. Then the third group we said was nocardiosis, and this is the one that occurs as an acute infection. In comparison to those chronic ones, it is something that could involve the lungs. So the causative agents of nocardiosis are species of nocardia. One of them is Nocardia asteroides. And the infection is acquired through inhalation. It can occur as a primary pulmonary disease, but is relatively more frequently encountered as an opportunistic infection of the lungs, affecting people with prolonged debilitating diseases. And it's associated with local spread within the lung and also distant spread through the various methods of like blood circulation, hematogenously or through the lymphatics. It has spread to other tissues. Somebody needs to mute. Who is that? So when it comes to investigations again, we just approaches generally. Specimens will depend on the manifestation. All in all, you have the discharge, you have the infected tissues, you have granules, something like nocardiosis, you probably have sputum, and then examination mainly involves demonstration of the organism in that specimen. And that can be done by staining and microscopic examination. Gram stain, we describe what they look like, and then modified Z0 insert staining for nocardia, is where you get acid fast organisms, but they are filamentous. Then culture is performed for isolation and identification. And we've mentioned relevant media for growth and suitable investigation environments. And then colonies again, we have talked about what kind of colonies one would get, the appearance, and when they're stained, again, microscopic morphology of the actinomycetes. And then there are some listed biochemical tests where there's difficulty in identification of enable one to get the species There are some relevant biochemical tests, like other biochemical tests in bacteriology. They are based on detecting certain enzymes and enzymatic activities of various substrates. And thirdly, in other laboratories, the infected tissue, part of it can be prepared, maybe fixed, and examined for filamentous bacteria. Then antimicrobial susceptibility of actinomycetes. Again, we are talking about different organisms, and their susceptibility will differ. And there are a wide range of antibacterial, antimicrobial agents which could be useful depending on what one is dealing with and what is available in that part. But among the antibiotics, penicillin is important and it's particularly effective for the causative agent of actinomycosis. Secondly, sulfur-containing agents. And the sulfur-diazine, if it's still available, then it can be used. And then the combination of sulfamethoxazole, trimethoprim, and other agents, these have been effective against actinomycotic mycetoma. There are also other agents which are not sulfur-related in that group. And thirdly, for the lower respiratory tract infection, the antibiotics like amikacin or minocycline have been effective, and also others against the causative agents of adenosis. So response to treatment is low, especially for the infections that have taken many years to develop. Also, treatment will take long because of the swelling and chronic inflammatory processes with fibrous tissue formation, which the antibiotic doesn't reach the site, the organism that easily. So treatment may take a long time, and that is important also to know whether it's going to respond or not. Sometimes it's hard to tell because it can take a long time before one sees the difference. But all in all, those that respond, they can take a long time, like 9 months to 12 months to be treated. Then there are some which are difficult to treat and require a combination of antimicrobial agents. And that, ladies and gentlemen, is the last slide for today's lecture. Are you still here? Am I talking to anybody?
Yes.
Then why are you not answering? Any remarks from you or questions?
It was a nice lecture.
Okay, so we stop there. We'll see you next time. I stop sharing. There we are.